Good morning. It's August 14th, and this is your daily brief in science. Here's everything you need to know. A solar eclipse drew lines of people across cities and coastlines as eye experts warned about the danger of looking directly at the sun and urged watchers to monitor for signs of solar retinopathy. Crowds gathered at major viewing spots and public events sold out as people queued to witness the celestial show. Totality appeared in parts of Spain, Portugal, and Iceland, while Germany recorded a substantial partial eclipse, underscoring the broad reach of the event. Gard Online carried multimedia coverage with an image and a comment section, tying the story to a wider eclipse reporting effort. Video thumbnails and related segments accompanied the piece, linking it to ongoing science journalism and audience engagement. News agencies and photographers documented observations from urban centers to coastal areas, capturing a range of perspectives on the phenomenon. A drone time-lapse is showcased as part of the broader effort to explain scientific events and support science journalism. The piece includes a retrospective with 12 images offering different viewpoints of the eclipse. Andrew McCarthy highlights space photography, including a capture named Shadows on the Balearic Sea, described as the moment the moon's shadow split the sunset. Photos feature locations such as the Javalambra Observatory and Hallgrímskirkja, illustrating the intersection of science and public interest. Reported eclipse coverage shows high visibility in major cities, with Plymouth around 94%, Cardiff about 93%, and Belfast near 93% at specific times. The report includes a contribution from La Presse reporter Daniele Solavagione, dated August 12, 2026. A mouse study on artificial hibernation shows memory can persist or even improve despite major loss of hippocampal connections and a drop in overall brain activity. Researchers found memory retention may rely on clustered engram synapses and the broader network architecture, not on every individual synapse. The findings challenge the idea that long-term memory depends on stable, persistent synapses across the brain. Instead, memories appear to be stored in resilient patterns of connected synapses that withstand large-scale remodeling. The study reports that dendritic spine size does not predict memory retention, shifting focus away from single synapses as memory carriers. This work suggests memories endure through selective architectural features, potentially guiding reconstruction of networks after disruption. First author Yuju Lin emphasizes the unexpected preservation of memory amid widespread synapse loss, pointing to a selective, clustered basis rather than uniform persistence. Researchers identified preserved motifs or clusters of connected synapses that survive hibernation and may steer the reassembly of functional networks. The team plans to map molecular pathways that support clustered engram synapses and investigate how non-clustered synapses reappear, with an eye toward drug targets to slow memory decline in amnesia or Alzheimer's disease. Published in Science, the study argues that long-term memory relies on the architecture of engram synapses rather than universal strengthening of all synapses. The researchers also note artificial hibernation could become a powerful tool to probe memory networks across mammals, including implications for humans, while raising questions about causality and how core memory clusters stay protected over time. A cross-organ, multi-model atlas of hypertension maps how cellular and molecular changes play out across 6 tissues, from the hypothalamus to blood cells. Using 3 animal models, the study juxtaposes angiotensin II-treated mice, Dahl salt-sensitive rats, and spontaneously hypertensive rats to reveal both shared and model-specific mechanisms of high blood pressure and organ injury. In each tissue, researchers charted cell type programs and gene activity to build a unified view of hypertension biology. The 6 tissues examined are the hypothalamus, kidney, mesenteric artery, left ventricle, middle cerebral artery, and peripheral blood mononuclear cells. A key finding is a conserved vascular smooth muscle cell gene program linked to blood pressure across models and tissues, highlighting a common vascular thread. They also identify cross-segment renal tubular programs tied to renal injury and blood pressure, and tissue-specific endothelial adaptations involving lipid handling, responses to shear stress, and cardioprotective signaling. Integration with human genome-wide association study data shows both model-specific and shared cell-type trait associations, indicating that each model captures complementary aspects of human hypertension genetics. Functional validation focuses on a non-coding variant, 28,451,064 rupees, demonstrating via genome editing its in vivo effect on blood pressure and regulation of nearby genes, such as SLC5A3 and RCAN1, potentially through chromatin interactions. The work emphasizes how multi-tissue, multi-model analysis can illuminate the complex genetics of hypertension and end organ damage. A new look at cancer signaling shows that neural activity inside tumors is more than a background hum. It shapes the immune landscape and the success of therapies. Sensory neurons and signaling molecules like CGRP, NGF, and TRPV1 influence how immune cells infiltrate tumors, drive T-cell exhaustion, affect NK cell activity, and push myeloid cells toward states that can support or hinder tumor growth. These neuroimmune interactions help explain why some tumors respond to treatments like PD-1 blockade while others do not, and they point to new angles for therapy. Researchers are exploring strategies that interrupt these neural signals to boost immunotherapy across cancer types, including blocking CGRP ramp1, targeting ITGA5, NGF neural remodeling pathways, and modulating TRPV1 and sympathetic output. Peripheral nerves and brain circuits coordinate both local tumor microenvironment changes and systemic immune responses, with stress and daily rhythms shaping anti-tumor immunity through autonomic control. The sympathetic nervous system, via beta-adrenergic signaling and catecholamines, influences tumor metabolism, blood vessels, the metastatic niche, and the function of immune cells, thereby affecting immunotherapy efficacy. Neuroimmune feedback loops can also drive resistance, as seen with perineural invasion in colorectal cancer and glioma-associated glutamatergic signaling that creates an immunosuppressive environment. Targeting these pathways could restore immune surveillance and improve outcomes in multiple cancers. This line of research ties together innervation, neurotransmitter signaling, glial activity, and neuroendocrine stress pathways as a multi-level framework for understanding and enhancing cancer immunotherapy. A new science study on the Paleocene-Eocene thermal maximum shows forests thickening first, then thinning as temperatures rose rapidly, leaving long-lasting reductions in canopy density and carbon storage that echo today's browning trends. In the PETM, forests opened up and large trees declined, altering climate, nutrient cycling, weathering, and animal habitats, a pattern researchers say mirrors modern forest stress and canopy loss. The study warns that if warming continues and is worsened by deforestation and wildfires, forests could sustain reduced carbon uptake and storage, strengthening climate feedbacks. Despite deep disruptions, floral composition and herbivory largely rebounded after the event, indicating resilience and a move back toward pre-PETM states. Forest recovery after the PTM took more than 100,000 years, with forests eventually denser than before as weathering drew down atmospheric carbon and helped rebalance climate. Researchers reconstructed the PTM-era canopy by analyzing fossil leaf cuticles to quantify leaf area index, providing a quantitative window into past canopy density. In Wyoming's Hanna Basin, a once dense forest of metasequoia, palms, and broadleaf trees contrasts with today's sagebrush landscape. Scientists linked leaf epidermal cell shapes to leaf area index to reconstruct past canopy density during the PETM from fossilized leaves collected in Wyoming. Takeaway on timescales. Forest recovery after extreme warming requires thousands of generations, highlighting the importance of avoiding climate thresholds that could trigger irreversible changes. The PETM involved multi-species ecosystem shifts, showing how climate stress can reorganize whole forests and their carbon-regulating roles. The loss of canopy during the PETM disrupted rainfall interception, soil stability, sediment transport, and broader environmental dynamics across landscapes. There are signs of large-scale reforestation today, but full recovery to pre-warming carbon levels could take up to 100,000 years, underscoring the urgency to curb warming. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.